Hello everyone, welcome you all to Sri Jain Rank Zone classes. I am your mentor and your friend for the subject biology, and I will be taking the zoology part, that is animal science. My dear students, in the previous videos we learn about the digestion, the steps of digestion in oral cavity, in gastric means the stomach, and we were discussing the digestion of food in small intestine, and now we will discuss in the coming videos for large intestine. Large intestine is basically meant for the absorption of food. Digestion, today we are going to start with digestion in jejunum and ileum. The hormones which are secreted in the duodenum, they stimulate the crypts of lubricant to secrete succus entericus, which is intestinal juice. This succus entericus mainly contains 99% of water and digestive enzymes less than 1%. Intestinal juice act on food. Succus entericus mainly contains the following enzymes: peptidase or erepsin. This is the type of exopeptidase. It converts oligopeptides into amino acids. Succus entericus mainly contains following enzymes. First is peptidase or erepsin. It is a type of exopeptidase which converts oligopeptides into amino acids. Disaccharidases. Disaccharidases, it includes sucrase. It is also known as invertase. It converts sucrose into glucose and fructose. Maltase, it converts maltose sugar into glucose molecules, into two glucose molecules. So maltose in the presence of maltase forms glucose plus glucose, and sucrose in the presence of sucrase or invertase, glucose plus fructose, right? Lactase, this enzyme is found only in mammals. It converts milk sugar lactose into glucose plus galactose. The lactose in the presence of lactase converts into glucose plus galactose. Intestinal lipases, this fat digestive enzyme converts fats into fatty acids and glycerol. Intestinal lipase, fatty acids and glycerol. Nucleotidase and nucleosidase, these act in the following way. So these, the hormones which are secreted at the starting of the duodenum, it stimulates the crypts of lubricant to secrete succus entericus or intestinal juice. This succus entericus mainly contains water 99%, and less than 1% it is having enzymes. Succus entericus mainly contains the following enzymes. First is peptidase, which is an exopeptidase which converts oligopeptides into amino acids. Then disaccharidases, which includes sucrase, which converts sucrose into glucose plus fructose. Sucrase is also known as invertase. And then maltase, which converts maltose into two glucose molecules. Lactase, which converts lactose, that is milk sugar, into glucose plus galactose. One of the most important thing, that lactase is only found in mammals. Intestinal lipase, this fat digestive enzymes converts fats into fatty acids and glycerol. Okay, nucleotidase and nucleosidase, they act in the following ways. These are DNases. DNases. Okay, nucleotides in the presence of nucleotidase converts into nucleoside and phosphate group. Okay, nucleosides in the form of nucleosidase converts into pentose and nitrogen base. This is nucleosidase. Okay, to nucleotide convert nucleotides into nucleoside plus phosphate, and nucleosidase converts nucleoside into pentose plus nitrogen base. Maximum digestion of carbohydrates is done in duodenum, but its digestion is completed in jejunum. Maximum digestion of carbohydrates is done in duodenum, but its digestion is completed in jejunum. Okay, is that clear? In herbivores, the symbiotic bacteria and the protozoans present in the cecum help in the digestion of cellulose. Very importantly, in herbivores, the symbiotic bacteria and protozoans present in the cecum, they help in the digestion of cellulose into glucose. So the digestion of cellulose takes place in the cecum by the process of decomposition. Okay, by the process of decomposition. Okay, yeah. So sorry. In herbivores, the symbiotic bacteria and protozoans present in the cecum help in the digestion of cellulose into glucose. As you know, cellulose is the polymer of glucose. Digestion of cellulose takes place in the cecum by the process of decomposition. This decomposition process is very slow, so very less amount of cellulose is digested at a time in cecum. In the last part of the large intestine, feces is temporarily stored. In the large in the last part of the large intestine, feces is temporarily stored. Okay. So there is very less amount of cellulose which is digested in cecum. Maximum digestion of food duodenum, while digestion of food complete in jejunum. Maximum absorption of food in jejunum. Millions of microscopic folds are present. These are finger-like projections which are present in the lumen of the gut, which are known as what? Villi. Okay. These villi increases the absorptive surface area, increases the absorptive surface area. Okay. Now, the villi are supplied with a network of blood capillaries and lymphatic vessels, largest of which is central lacteal. These cells that line the surface of villi, numerous microscopic bristle-like projections, they are known as what? Microvilli or brush border. Okay. The villi are supplied by a network of blood capillaries and lymphatic vessels. The most important is central lacteal. The cells that line the surface of villi, numerous microscopic bristle-like projections are present, which are known as what? Microvilli or the brush border. That is why it is known as what? Brush border epithelium. Yeah. Next, the further increase the surface area for the absorption of the nutrients digested food on the surface of mucosal epithelium are billions of single cell mucus glands called as mucus or goblet cells. The mucus cells or goblet cells are also present, which are present on the surface of mucosal epithelium. These help in the production of mucus, and with the help of villi, they help in the absorption of food. Okay. The cells mainly secrete mucus that acts as a lubricant. They act as a lubricant and protects the epithelial surface for damage and digestion. Okay. The cells mainly secrete mucus that act as a lubricant and protects the epithelial surface from damage and digestion. Okay. Now, milk sugar, it remains undigested due to the insufficient lactase. Okay, in case of when we talk about humans, okay, so the milk sugar remains undigested because of insufficient lactase. It is digested, but it is partially digested. Okay, that is why to digest milk, okay, or milk is supposed to be, digested slowly. Okay, so that is why it is, and that is why it is considered or it is recommended to take milk, but not to take any other thing apart from it. Okay, because the digestion process of milk sugar takes long time. These are villi. The villi, they are the long finger-like projections. You can see the folds over here. These are the intestinal villi. Okay, these are the submucosal layers, then serosal layers, inner circular longitudinal muscle layers, muscularis layer, capillary network. You can see goblet cells. lacteals, central lacteal, intestinal glands, lymphatic nodules, enteroendocrinal cells secrete hormones, simple columnar epithelial and microvilli absorbs nutrients. Okay, so these are finger-like projections known as what? Villi. Epithelial surface showing absorption of nutrients, the lumen of small intestine, proteins to amino acids, carbohydrates to monosaccharides, bile salts to fat globules. Okay, then emulsified droplets, free fatty acids, monoglycerides, microvilli, they absorb all these nutrients, epithelial cells of the small intestine. Okay, they absorb everything, smooth endoplasmic reticulum, chylomicrons, lymphatic capillary lacteals, okay, blood capillaries. Blood capillaries, they mainly carry amino acids and monosaccharides, and lymphatic capillary lacteals, they mainly carry chylomicrons, which are the we can say they are the fat droplets, monoglyceride droplets, okay, along with the proteins. These are chylomicrons. Now, before moving to this, I would like to add something more. Yeah, here. Digestion of cellulose. It remains undigested in humans. It acts as a roughage or a fiber. Cellulase is absent in vertebrates. Absent in vertebrates. Ruminants have symbiotic microorganisms. They have symbiotic microorganisms. In their gut, which aid in cellular digestion. Which help in cellulose digestion. Okay. In horse, donkey, the microbial fermentation occurs in cecum. Basically, microbial fermentation occurs in cecum. Horse, donkey. Okay. Yeah. Let's move to the next slide. Role of hormones. Role of some major gastrointestinal peptide hormones in digestion. Gastrin. The source is pyloric stomach and duodenal G cells. Vagus nerve activity. Peptides and proteins in stomach. The stimulus is vagus nerve activity. Okay. Vagus nerve basically it initiates or stimulates the parasympathetic nervous system to release acetylcholine. Vagus nerve activity, peptides and proteins in stomach, they are the stimulus to secrete gastrin. Target secretory cells and muscles of the stomach, secretion of HCl and stimulation of gastric mobility. The action and the targets are secretory cells and the muscles of stomach and the action is secretion of HCl and stimulation of gastric mobility. Cholecystokinin, the source of secretion is upper small intestine, that is duodenum. Stimulus is food, fatty chain, amino acids in duodenum. Target is gallbladder, and action is contraction of gallbladder to release bile. Secretin, secreted from intestinal wall, duodenal wall. Stimulus is food and strong acid in stomach and intestine. Target is pancreas. Action is to secrete water and bicarbonate ions and inhibition of gastric mobility. Okay. Targets are pancreas, secretory cells, and muscles of stomach. Gastric inhibitory peptide, upper small intestine, duodenum. Stimulus is monosaccharides and fats in duodenum. Target is gastric mucosa and muscles. Action is inhibition of gastric juices, okay, and mobility, gastric mobility, showing food passage. Okay, slowing food passage. Sorry. The gastric inhibitory peptide, it slows down the food passage. Okay, gastric inhibitory peptide is anti-gastrin. Is it decreases the release of gastrin. So these are the hormones: gastrin, cholecystokinin, secretin, and gastric inhibitory peptide. These are the major gastrointestinal peptide hormones required for digestion. Understood? Absorption of food. The lining cells of villi are columnar epithelial cells called enterocytes. On the surface of enterocytes, numerous microvilli are found. They increase the surface area of mucous membrane. The lining cells of the villi, they are columnar epithelial cells and they are known as what? Enterocytes. On the surface of enterocytes, numerous microvilli are found. They increase the absorptive surface area. Absorption in buccal cavity. No absorption of food takes place in the oral and esophageal cavity. Some only some mechanical and chemical only some chemical medicines and alcohol are absorbed in oral pharyngeal cavity. Absorption in stomach. In the stomach, absorption of water, small salts, alcohol, glucose takes place. Complete absorption of alcohol takes place in stomach. Complete absorption of alcohol takes place in stomach. Absorption in duodenum. Iron, calcium are absorbed in duodenum. Absorption in jejunum. Maximum absorption takes place in jejunum. Iron and calcium, they are absorbed in duodenum. In stomach, absorption of water, some salts, alcohol, and glucose takes place. Complete absorption of alcohol takes place in stomach. Okay. No absorption of food takes place in oral and pharyngeal cavity. Okay. Carbohydrates. The principal carbohydrate of our body is usually starch from rice or wheat, which is broken down by pancreatic amylase. Disaccharides are broken down to their monosaccharide by enzymes succus entericus. Monosaccharides are absorbed by capillary blood within the finally reach to the portal vein. Absorption of glucose molecules occurs only along with sodium ions by active transport. Fructose is absorbed passively. Fructose is absorbed passively and glucose is absorbed by the active transport along with the sodium ions. Digestion and absorption of amino acid. All these proteins are exposed to pepsin, trypsin, chymotrypsin, carboxypeptidase, etc. And as a result, they are converted to tri and dipeptides or free amino acids. Amino acids are of two types: L amino acid and D amino acids. L amino acids are naturally occurring. 7 are absorbed by active process against the concentration gradient, while D amino acids are absorbed passively by diffusion. So L amino acids, they are naturally occurring and they are absorbed by active process against the concentration gradient, while the D amino acids, they are absorbed passively by diffusion. Di and tripeptide enter the enterocytes where they are hydrolyzed to amino acids by dipeptidases and then absorbed by portal blood. So di and tripeptide, they enter the enterocytes where they are hydrolyzed to amino acids by dipeptidases and then absorbed by portal blood. Is that understood? The principal carbohydrate of our food is starch, which is broken down by pancreatic amylase. Disaccharidases secreted by succus entericus of intestinal juice. They convert disaccharides into monosaccharides, and monosaccharides further converts or absorbs into the portal vein. Absorption of glucose molecules occurs only along with sodium ions by active transport. Fructose is absorbed passively. Fructose is absorbed passively. Digestion and absorption of amino acid. All these proteins are exposed to pepsin, trypsin, chymotrypsin, carboxypeptidases, and as a result, they are converted to tri and dipeptides or free amino acids. Amino acids are of two types: L amino acid, D amino acid. L amino acid actively absorption, D amino acid passive absorption. Di and tripeptides enter the enterocytes where they are hydrolyzed to amino acids, broken down into amino acids by dipeptidases, and then absorbed by portal blood. Okay. Digestion and absorption of fat. One molecule of triglyceride is hydrolyzed into one molecule of monoglyceride and two molecules of fatty acids by pancreatic lipase. Pancreatic lipase convert one molecule of triglyceride into one molecule of monoglyceride and two molecule of fatty acids. After hydrolysis, the bile salt, monoglyceride, and the fatty acid together produce a complex called mixed michel. These are water soluble and enter into the enterocytes. Monoglyceride and fatty acid are resynthesized within enterocytes to form molecule of triglyceride. Triglyceride combines with small amount of protein and resultant complex is called chylomicron. 150 microns size, white in color. Chylomicron enters the lacteal. Fat soluble vitamins are absorbed along with the dietary fat, whereas water soluble vitamins are absorbed by passive diffusion. Vitamin B12 is absorbed with intrinsic factor by forming a complex. In ileum, vitamin B12 and bile salt are absorbed. In colon, only water is absorbed. In colon, only water is absorbed. In ileum, vitamin B12 and bile salts are also absorbed. Okay. Fat soluble vitamins are absorbed along with the dietary fat, whereas water soluble vitamins are absorbed by passive diffusion. Vitamin B12 is absorbed with intrinsic factor, forming a complex. Monoglycerides and fatty acids are resynthesized within enterocytes to form a molecule of triglycerides. Triglyceride combines with a small amount of proteins and resultant complex is called chylomicrons. Chylomicron enters the lacteals for the further absorption. After hydrolysis, the bile salt, monoglyceride, and fatty acids they produce a complex called mixed michel. Mixed michel enter the enterocytes. All lymph capillaries coming out of the alimentary canal unite to form lymph vessels. All lymph vessels coming from alimentary canal into left thoracic lymph duct. This duct now opens into the left subclavian vein. Through the blood, fat reaches the heart and form. Here it is distributed throughout the body. Beside fats, other substances of the digested food like sugar, amino acids, vitamins, minerals, salts, after being absorbed, enter the blood capillaries. All blood capillaries coming out of the alimentary canal. All lymph capillaries coming out of the alimentary canal unite to form lymph vessels. All lymph vessels coming from the alimentary canal into the left thoracic lymph duct, which opens into the left subclavian vein. Okay. Besides fat, other substances of the digested food like sugars, amino acids, vitamins, minerals, salts, after being absorbed, enter the blood capillaries. All blood capillaries coming out of the alimentary canal join together to form hepatic portal vein. This is hepatic portal vein. This vein takes the digested food material into the liver. From the liver, the hepatic vein and the superior post-caval vein takes to the heart. Heart distributes them throughout the body. Liver performs some necessary and important actions on the digested food. So this is the distribution, how absorption of or distribution of nutrients to the whole body takes place. Okay, is that understood? I hope it is clear. This portal vein, hepatic portal vein, and the superior post-caval vein takes to the heart. Heart distributes them throughout the body. Liver performs some necessary and important actions on the digested food. So that will be all for today. We will be meeting in the next turn to further carry on with the digestion, so absorption process, and we will discuss the rumination and the different parts of ruminant's stomach. Till then, bye bye. Take care. Stay safe. Stay healthy. Stay at home.